All right. All speakers present and accounted for. Anybody here know about Calypsy? Oh, all right then. Well, so I'm Doug Johnson. I'm the director of California Invasive Plant Council, and
I'm Jutta Berger. I'm a science program director with Calypsy.
And we're happy to be here at CNPS, sharing some of the content that often gets shared at the Calypsy Symposium and integrating the two. So today we have 4 excellent speakers on talks that cover research that guides our understanding of the impacts of invasive plants, our understanding of the best ways to control invasive plants, and may even lead to policy that helps support control of invasive plants. Right, Stu? Right. Excellent. Do you want to say anything else about the session?
I think we're taking the whole gamut from really interesting research that's going on to some great success stories on application and treatment control of invasive plants. And this is a 2-part deal. We have another session tomorrow as well. It's session 27. So our first speaker will be doing a double session, a 40-minute presentation, and then we'll be going to our standard 20 minutes. Not quite 40 minutes anymore.
Right, not quite.
It's a double whopper. Well, let me introduce this guy, Dr. Stuart Weiss, who got his degree from Stanford, is chief scientist at Creekside Science, which is a small consulting firm that brings cutting-edge science to biodiversity conservation. Creekside Science pursues projects ranging from restoration of endangered plants and butterflies to regional conservation planning, nitrogen deposition, which we're going to hear about, and climate change. And we are here in the Santa Clara Valley, where Stu has had an impact on the policy and the funding of biodiversity protection. So he's here to tell us about it. Stu.
Okay, thanks. How many of you have heard about atmospheric nitrogen deposition? How many of you have heard about it from me? Okay, we need to fix that. Okay, so I'm going to give you the take-homes up front. Smog acts as nitrogen fertilizer. Nitrogen increases the growth of annual grasses. Annual grasses are the most pervasive invasive threat to imperiled California flora. I think we can be pretty solid about that. And it's through direct competition, it's through thatch buildup and fire cycles. So we have managed grazing or precision mowing or practical management solutions, depending on what scale you're at. A high fraction of rare, threatened, and endangered taxa are exposed to high nitrogen deposition and at risk. And nitrogen deposition is not a standard subject of environmental review. You rarely see it brought up in CEQA, but it's something that comes under the cumulative impacts. There are regulatory precedents. I'll tell you about some of them. And let's get nitrogen deposition impacts into CEQA review and ESA permitting. And then we can generate desperately needed stewardship resources. So nitrogen is N, really big. And it's the biggest global environmental change that almost nobody has ever heard of. And it all has to do with the reactive forms of nitrogen. 78% of the atmosphere around us is nitrogen gas in the form of N2, which is very unreactive because of a very strong triple bond. But then when it gets into reactive form, then it starts doing its thing. So if we look at what they call planetary boundaries, if we look down in the biogeochemical flows, you see the red wedge there of nitrogen. So the nitrogen cycle on a global scale is more disrupted than any other biogeochemical cycle, and it has really pervasive effects ranging from coastal eutrophication, the dead zone at the mouth of the Mississippi River, Chesapeake Bay, and it leads to terrestrial eutrophication, i.e. nutrient enrichment that changes the nature of terrestrial plant communities. So A lot of different sources. There's combustion, there's vehicles. Vehicles both produce nitrogen oxides and ammonia because of the catalytic converters we put on them. A lot of sources in agriculture, direct application of fertilizer, gaseous emissions and runoff, and then concentrated animal agriculture. Then there are the natural emission sources. There's lightning, fire, and then soil processes. So I'm not going to go into the nitrogen cycle too much because it is fiendishly complex. So this is the site a lot of us have seen in California. This is south of San Jose looking up towards Silicon Valley, that brown cloud. There's a process called dry nitrogen deposition, and basically smog is nitrogen fertilizer because of this deposition process. So the big three compounds are nitrogen oxides, that's NO and NO2, and that's what comes out of direct emission from combustions in soils. Then nitric acid vapor in the air is a reaction product of the nitrogen oxides. And then we have ammonia, which is emissions from fertilizer, animal waste, and vehicles, and other sources. So deposition takes various forms. There's wet deposition, which I think a lot of people think of like acid rain. That's nitrogen in the form of nitrate and ammonium coming down in rain, snow, and hail. Then there's dry deposition, which is the gases are either being absorbed directly into the plant stomata when they're active, or they're sticking to surfaces and getting washed into the soil. So it's a pretty complicated process, but fortunately people have figured it out largely. So again, dry deposition is atmospheric particles and gases depositing directly to the plant soil surface, and stomatal absorption, and then wet deposition. And then in California, dry deposition dominates, except in the Sierra Nevada where we get very heavy snowpack. So you'll be quizzed on this one. No. The ecosystem's effects of nitrogen deposition. Just remember, most ecosystems are fundamentally limited by nitrogen. So the productivity, and then when you start adding nitrogen to the system, you start seeing this cascading effect. So it's fiendishly complex. We're still discovering new pathways for nitrogen. But in the end, what it does is it juices up the rate of nitrogen cycling. So you're not just seeing the nitrogen availability that's coming from this year's deposition, you're seeing this cumulative impact of speeded up nitrogen cycling that makes it more available for plants. So we got to think of this in terms of a cumulative dose, not just this year's deposition. So what does nitrogen deposition do? Makes annual grasses grow really strongly. So this is a cornfield. That's an annual grass. We put large amounts of nitrogen to make it grow 8 feet tall. Here's wheat. Same thing. We feed it lots of nitrogen with sufficient water. It grows really well. Rice, same thing. Then in nutrient-poor systems, this is a serpentine grassland about 10 miles south of here on Tulare Hill. This is kind of the natural serpentine grassland, incredibly beautiful, diverse ecosystem. But under atmospheric nitrogen deposition, somewhere on the order of 15 to 20 pounds per acre per year in this area, this is what it turns into. It's turned into this because the cattle were removed from this site. It's a long story, I can't get into it now. But again, nitrogen makes the annual grass grow. It grows so strongly it crowds out the little native wildflowers, and then we get this buildup of thatch, which smothers the system. So it's not just serpentine grassland. Here's some grasslands on San Bruno Mountain, which is surrounded by urbanization. We have very, very strong growth of annual grasses. There's my former staff member, Lek Naumovich, who many of you know, standing in a stand of primarily wild oats. but very few native forbs in that grassland, especially on the lower slopes of San Bruno Mountain. Higher on the mountain, we have these gorgeous, gorgeous coastal prairies filled with things like viola, lupins, or the endangered butterflies on San Bruno Mountain have host plants. And highly recommend going there in the spring along the main crest of San Bruno Mountain, because it's high enough up that it's getting clean ocean air. It's not getting the smog so much from the urban areas around it. Vernal pools. So we have a lot of vernal pools, like in the Central Valley, which, as you'll see, is a very high nitrogen deposition area. So it's another one of those circumstances where when it's being grazed appropriately, there's plenty of room for the little forbs, little annual forbs to, grow and thrive. But with the nitrogen deposition, we get this excessive growth of annual grasses that actually dry out the pools if you let it go on for too long. So it's not just crowding out and thatching over, it's literally changing the hydrology. So here's an ecological reserve up in the Santa Rosa Plain set aside for vernal pools. Well, it's not being grazed, it's not being managed, so that it's totally overgrown. It turns out the Santa Rosa Plain is a high nitrogen deposition zone. So then we have coastal sage scrub. This is where a lot of the original work was done by Edie Allen and her students and colleagues at UC Riverside. So, coastal sage scrub, lots of room in between the plants, lots of beautiful wildflowers, but it happens to be close to LA and San Diego. So we end up with the shrubs get burned up, and it ends up being kind of a weedy annual grassland. So we're losing like these large swaths of land to this phenomenon. The desert is a really interesting place. It makes the annual grass grow. I'm sorry to keep beating on that, but that's kind of you get red brome and you get Mediterranean split grass. So now we have a fine fuel load that's continuous that doesn't break up. So now fires carry across the Mojave Desert where they never would before because of this nitrogen enrichment. So a lot of ecosystems are being affected, especially the low biomass, nutrient poor systems. So there's a synthesis paper in 2010 that reviewed all the evidence and set up what are called critical loads. I'll talk a little bit about that. But this is a really good source for seeing what the research was up until 2010. And now even, there's been like a decade more work done on it. So, let's kind of sum up the ecological impacts of nitrogen deposition here. We have urbanization and agriculture leads to nitrogen deposition. The nitrogen deposition juices up the annual grass growth, and then also with the disturbance, we're getting more exotic species taking place. The grasses outcompete, but also they create a fire cycle in places that normally wouldn't be able to burn that frequently. And that leads to really profound changes in the, I got music next door. And it changes the fire cycle. Okay, so it's really complicated, but some very smart, talented people have built a model called TDEP, total deposition, built by folks at the EPA. And it's a fusion of measurement and atmospheric modeling. It's a really good synthesis. And across the United States, they have dry deposition modeled at 12 kilometers, and then wet deposition at 4 kilometers. And it's annual data from 2000 to the present. They lag about a year behind. And it breaks it down by different nitrogen species. So you can see how much nitric acid vapor, how much ammonia, how much particulate nitrate, how much, et cetera, et cetera. So you can really pick apart the nitrogen. So you can see the eastern United States and the Midwest, really nitrogen deposition hotspots. And then you can see California there. We're going to take a closer look at California now. So here's the map of California. Locate yourself on that. How many people are in the red zone? Okay. How many people are in the slightly less red zone? How many people are in the yellow zone? How many of you fortunate people are in the dark blue zone? Okay, so it's all over the place. So we need to keep this in mind when we're trying to manage habitats, that there's this forcing factor that's external to your system that's making the annual grass and other weeds grow more vigorously. So, we have the urban areas. There's the Bay Area, Southern California, Sacramento, real hot spots. Then we have the agricultural areas, the Central Valley, Sacramento Valley, the Imperial Valley. And then if you look closely, you can see these plumes coming out of these areas. So the downwind areas like the Sierra foothills and up towards Lake Tahoe are getting nitrogen blowing in from upwind areas. Southern Sierra Nevada is a really large, really major impact area. We have the maps here. So we can also start breaking it down by, is it oxidized nitrogen or reduced nitrogen? They have different properties. Oxidized comes from nitrogen oxides, from combustion and then soil emissions. Reduced nitrogen is from ammonia emissions from agriculture, animal agriculture in particular, but also vehicles are producing ammonia. That's why there's so much, that red spot in the LA basin there. And if you look really closely, you'll see there's a slightly pink spot here in San Jose. That's because of traffic. So, who's exposed? Overlay the map onto the CNDDB. So I did this several years ago, and we were using the nitrogen deposition averaged from 2014 to 2016. It's changed a little bit since then. And then I just lined them up from the most exposed species over there at 1 to the least exposed species at 26. And turns out that 62% of these threatened and endangered listed taxa in California are exposed to greater than 5 kilograms of nitrogen per hectare per year, which is where you start really seeing effects, especially in low biomass, nutrient-poor systems. And then you can see it's divvied up between oxidized and reduced nitrogen. Which is kind of a second-order component of it. It's the total nitrogen that's really the most important. But then within each of these taxa, there's a range of exposure. So here I'm just showing the same order, but the minimum and maximum in addition to the average. So you can see within some taxa, some occurrences are, not exposed to so much, but others are highly exposed. So it's really important to know exactly where you are on the nitrogen deposition map to understand what's going on with your taxa. Then we have the rare species. These are the unlisted species, but are rare enough to show up in CNDDB. 44% of these are exposed to greater than 5 kilograms of nitrogen per hectare per year. So the basic point here is we've got a real problem here, and very few people are talking about it. So we can use this analysis. So here's a regional scale analysis of the Santa Clara Valley Habitat Plan, which is a habitat conservation plan, natural communities conservation plan, where nitrogen was actually one of the major nexuses for it. So I just pulled out from that database what the mean, max, and min are of all of the CNDDB taxa in the area. This is a relatively simple analysis to do. So it's a really good place to start to see who's exposed and who's exposed to how much. Now, this is done at like a 4 to 12 kilometer scale, But we also have very localized effect. So here's Edgewood Natural Preserve up near Redwood City, about 15, 20 miles north of here. I actually measured nitrogen gases at these 3 sites, west of the freeway, east of the freeway, and about 400 meters east of the freeway. Generally, the prevailing winds are from the northwest, so we should be seeing a pretty good deposition gradient here. And when we look at these 3 sites, we see the highest site is just east of the freeway, where we're getting, this was in 2002, 2003, where about 15 kilograms per hectare per year. And for those, it's about 15 pounds per acre conveniently, so if you want to kind of scale things. But then you see it goes down to about 5 when you're 400 meters east of the freeway. But looking at like what gases is doing it, well, that's the ammonia. And it's like, well, where's the ammonia coming from? We'll get to that. So what's the impact on the ecosystem here? Did some plant transects there. So you see where Highway 280 is, and then meters from Highway 280, west versus east. And then this is the cover of 2 key species, Plantago erecta, host plant of the Bay checker spot butterfly, AKA the most important plant in the world. And then Italian ryegrass, Lolium, which is now Festuca perennis. And if you look at the Lolium curve here, you see there's a zone extending out about 300 meters from the freeway where it's like really dominant, over 30% cover. And then the Plantago is really suppressed, 3% or less. Then we get about 400 meters east of the freeway. It's kind of switches back and forth. So, you know, the effect goes out about 400 meters from a freeway. So this ammonia from cars is like a new thing in the environment since about 1990 when we put catalytic converters on. So catalytic converters are great at chemically reducing nitrogen oxides, but they over-reduce it to ammonia. So now cars are putting out about equal amounts of NOx and ammonia. And ammonia is a very, very potent deposition agent because of its chemistry. So that's Highway 280 there that has about 100,000 cars a day going by on it, slicing right through a beautiful serpentine grassland. Okay, so there are regulations. We have the Clean Air Act. Regulates nitrogen dioxides. There's an annual standard. Doesn't regulate ammonia, and it regulates NOx because NO2 is poisonous, and also NO2 is a ozone precursor, so it reacts with hydrocarbons in the atmosphere and creates ozone, so it's very heavily regulated. Air Resources Board in California has a lower NO2 standard, and some really interesting NOx market trading for new sources. I can't go into the details of that. It works. Regulation works. Here's the concentration of nitrogen dioxide just down the road here in San Jose. So you can see the annual average was about 0.3032 parts per million in 1988, and it's come down by about 2/3 to pretty close to 0.1, 0.2. So regulation works. So thank you, Air Resources Board and EPA. We can look at what are the trends statewide according to TDEP. So the blue areas are where we've seen substantial decreases in nitrogen. A lot of the urban areas, that's because cars have just been heavily regulated for NOx emissions. But we can see there are some red areas where nitrogen deposition has gone up a bit. Now, if we look at the NOx, regulated, ammonia not regulated. So NOx is down all across the board, especially in the urban areas. The ammonia is either holding steady or is going up because we've made this trade-off. Then We have the exposure trend. So what I'm graphing here is the exposure of these occurrences in 2000, 2002 versus 2014 to 2016. So you can see it's generally come down quite a bit in a lot of the, especially in the really high deposition areas, because again, the NOx is being regulated pretty effectively. So we have this tool that's been called a critical load, which is a quantitative estimate of an exposure to one or more pollutants below which significant harmful effects on specified sensitive elements of the environment do not occur according to present knowledge. But that's so qualified there. It's just, but it's, you know, it's a tool, because if you're in areas where you're above the critical load, you know you're going to be having some kind of impact. So the critical load is dependent on, you know, the duration, total amount, what form of nitrogen, the intrinsic sensitivity of the plant species present, and the abiotic conditions in the ecosystem. But the thing to remember, it's this cumulative process. When we start looking at nitrogen deposition impacts, it turns out you get better correlations with things like biodiversity loss with what the cumulative exposure has been over decades, as opposed to what the instantaneous exposure is in the recent years. So in that Fenn et al. 2010 paper, set critical loads across California for different ecosystem elements. The red areas are where you're really far exceeding the critical loads. A low end threshold exceedance usually has something to do with lichens. They're very, very sensitive to air pollution. And then in the high end, you know, you can see there's still large areas of the state, like the Sierra foothills. You know, I highly recommend getting that Fenn et al. Paper and perusing it. So, you know, here's serpentine grassland. On the right side of the fence, it's been grazed at typical levels, you know, 1 cow-calf per 10 to 15 acres. The cows were pulled off the left side, and it turned into a sea of Italian ryegrass. So what we've discovered here is that cattle are absolutely essential to maintaining the serpentine grassland in high deposition zones south of San Jose, and that's because cows eat grass. That's smog is fertilizer, cows eat grass. So those are my 3 words here. And it also means that we need to, the keystone species in the system are the ranchers. You're not going to have cows out there. You know, it's kind of this ecological web. Here's a, you know, co-adapted mammalian biodiversity complex here. I see 4 species there, yeah. All working to maintain the serpentine grassland. Little did they know it. So this is, you know, impacts like this have led to the formation of the California Rangeland Conservation Coalition, where, you know, we've discovered we have a lot in common with the ranching community. Another method is mowing, precision mowing. It can't be done at the scale of cattle grazing, but mowing passes. The right side here was, the right side here was mowed the previous year, so lots of flowers. The left side was the control, and it passes what I call the O test. Obvious. But it also passes the F test. Threw down some transects, and, you know, the mowed side had about 35% plantago, again, the most important plant in the world, and less than 5% ryegrass, and then the unmowed side had about 30% cover of the annual grass and about 10% plantago. So it really worked. It lasts for a while, too. So here's the bay checker spot butterfly. I had to show the bay checker spot butterfly here. But because of the nitrogen deposition impacts on this, here's the beautiful serpentine grassland. I kind of untangled the nitrogen story, published a paper in 1999. This became the best available science under the Endangered Species Act. So we started getting mitigation for big nitrogen sources like the Metcalf Energy Center, which is a gas-fired power plant. This led to 131 acres being permanently protected, an endowment, and then 30 years of operating expenses. We got 2 more power plants that led to another 80 acres, and these were pretty far away, so the cumulative impact aspect of it is really powerful. And then they wanted to widen Highway 101 down at the base of the hill there. We got 540 acres of that. For that as direct mitigation, then a commitment to a habitat conservation plan. I've talked about this as the banquet speaker and elsewhere. We just had that was in 2001. Over the next decade, we lobbied, Operation Flower Power. We brought elected officials up to see, bring the power to the flowers, as it may be. And by 2013, we had the signing ceremony of the implementation agreement of the Valley Habitat Plan, and nitrogen was the biggest nexus for this. So it's possible to do something with this. And this led to $665 million estimate over 50 years and conservation of about 42,000 acres. And there's going to be a big fat endowment at the end of it for management into perpetuity. Well, that's not supposed to be there. I'm continuing to do some work. We're finding that nitrate is seeping out of the serpentine grasslands in all the springs, up to 5 parts per million nitrate is nitrogen, which is half the drinking water standard. Basically, it's just flushing right through the system. It's so saturated. Coyote Ridge Open Space Preserve was like the big first acquisition under the Habitat Plan, primarily serpentine grassland. There's Matt Freeman of the Open Space Authority with cows on the brain. So this paper generated about $700 million worth of mitigation, which is like, yeah, I like that kind of impact. But there's so much more to do. And thankfully, the bay checker spot was an endangered animal, which, under ESA is really important. We had some more success at the Antioch Dunes, Lang's meadowlark butterfly and 2 beautiful endangered wildflowers, which actually it literally is a postage stamp of 55 acres, and the species do exist more than on the postage stamps. Here's the Antioch Dune evening primrose with Mount Diablo in the background. See some open sand there. Here's the Contra Costa wallflower. Again, lots of open sand. But a lot of the dunes looked like this, covered with annual grass. This accumulation of nitrogen at around 7 kilograms per hectare per year for decades just led to the annual grass just completely taking over the dunes. So there was another power plant project that was proposed out there. The Wild Equity Institute threatened to sue. We got like a $2 million settlement for this power plant, but they never built the power plant, so it's probably a good thing. But it is possible to use the law to get mitigation for these things. You can do a regional scale analysis. Here's Southern California, the nitrogen deposition total, average annual for 2014 to 2016. You can just see the LA basin, but you can see the plumes going out into the desert. See San Diego is also a hot spot. We can overlay this onto conservation areas. So here's nitrogen deposition overlaid onto the Keno checker spot recovery units. So we can see down near San Diego, the southwest one is pretty high nitrogen deposition. And you can totally see it out there, except on the clay-rich lenses, you don't have the Plantago erecta growing. It's all dominated by annual grasses. And then up the closer you get to the LA basin up in Riverside, really high deposition as well. So you can do these analyses and go, if there's a CEQA process, if there's some conservation plan, you just say, hey, you have a nitrogen deposition problem. Here's the analysis. So here's my call to action. We have the best available science on the process. We need to kind of update the nitrogen deposition exposure and look at the habitats of the different species and document the threats to specific taxa. We need to get persistent CEQA comments going. If we keep bringing it up under CEQA and then perhaps sue over it, then it will get into CEQA. We have to realize it's cumulative impacts. So it's not, any one project is not going to be what's driving you over the edge, but there is a cumulative impacts approach to things like this. You know, lawsuits like we did at Antioch or threatened to at Antioch. And we got to get the wildlife agencies to internalize this and realize that it's happening, and that's up to a lot of us to make them aware. I'm working with CDFW and U.S. Fish and Wildlife Service, but we need many more voices on this. And then the mitigation, we need stewardship resources on existing protected lands. We're not going to ask for like huge acreages in addition to what we already have. We have to take care of the lands that we have. And this nitrogen deposition is really threatening a lot of the biodiversity on those lands. So maybe some success raising the butterfly nets over Coyote Ridge. We did it. But, you know, just to kind of carry the Iwo Jima analogy a little further, raising the flag over Mount Suribachi was only the beginning. They had an incredible battle for weeks after that. So it's not done yet. This is like we're having some initial success. So thank you. Oh, great. Surely there must be a question. Way in the back. First, okay, yeah, okay, the question, the first part of the question, which I thank you for translating it, was about electric vehicles. Yes. There are, given the current mix of electricity, there are indirect emissions from the gas-fired power plants, but it's much more efficient, much less NOx and ammonia production per unit for mile traveled. So yeah, electric vehicles are going to do a really help. The balance with the cattle industry, it's like, yeah, there's a lot, you know, there's always trade-offs. And, you know, if we did not have the cattle on the serpentine grasslands on Coyote Ridge or in our vernal pools, we would lose those species in a matter of just a few years. We're not going to reform the food system, and we're kind of stuck with what we have there. So on balance, I'd say we need the cattle there. You know, grass-fed beef is one small niche that might be good, but we need to have something out there taking the grass away.
One more question.
I support cattle. Yeah, I support ranching as well in a sustainable way. And to kind of dovetail on that, I'm curious, were there any studies in your area that looked at the nitrogen, oh my gosh, okay, sorry, that looked at the nitrogen that was deposited through the cattle on that area compared to the nitrogen?
Okay, yeah. So I managed to get this into the Cars, Cows, and Checker Spot papers, that cows eat globally but deposit locally. there's a net export of nitrogen from the system from the cattle because they're growing. The cattle are really after nitrogen. They're not after just kind of biomass. So it turns into protein and gets exported from the system. It's not like a huge amount, but it's definitely a net export from the system. The impacts of cow pies are transient over a few years. I sort of have this image of a study I call the biogeochemical demography of cow pies, where it gets deposited, starts decomposing, grasses grow up really lush around it. Cattle don't touch it because it's still like feces there. They're smart enough to not eat their own feces. But then eventually there's a really nice, rich patch of grass, and then they really go after it. And then after about 5 or 6 years, it's pretty much gone. The urine patches tend to be a little more persistent because it's a much richer source. So, that it? Okay, thanks.
Thanks, Stu, for a really inspiring talk. It's great to hear the policy success story and to see how research can really feed into making policy and helping to inform control. So moving on to our next speaker, Marina LaForgia. I am very happy to introduce Marina, who is coming to us as a recent PhD graduate from UC Davis, and she's now a postdoctoral fellow as well at UC Davis and at the University of Oregon. She also is near and dear to Calypsy's heart because she was an excellent student liaison to our board of directors a few years back. She studies how plant communities cope with environmental variability and climate change, and is particularly interested in how seed and seedling stages inform this response. And that's what she's going to be talking to us today about.
All right. Well, thank you all for coming, and thanks, Jutta, for that introduction. It's so lovely to be together again after so long. Today I'm going to be talking about some preliminary results from a project I'm working on that uses seeds and their traits to help us understand species spread, seed banks, and potential for invasiveness. So I work predominantly in annual grasslands. These systems make up about 10% of California here in Orange, but they are habitat to something like 40% of our native and endemic plant species, and 90% of our listed plants. These systems also provide critical resources for our pollinators and are incredibly good at storing carbon below ground. Grasslands are also California's most altered ecosystem. The majority of our grasslands are dominated by a low number of invasive annuals from Eurasia, like lolium that Stu was talking about, goatgrass, fillory, medusa head, and yellow star thistle. These invaders grow really quickly and typically have high reproductive rates that allow them to rapidly increase in number. Native species, on the other hand, make up a smaller but more diverse part of this community. Natives are notoriously seed limited compared to the invasives. Not only that, but they can be really difficult to get them to germinate, which can make restoration of these species back into the community very costly. Some of these difficulties with native species is due to their seed banks. So California is extremely temporally variable. No 2 years seem to be exactly alike. And to cope with this, many species have evolved seed banks where their seeds survive for years below ground before germinating. And a healthy native seed bank can be a buffer towards disturbances and environmental changes, allowing native populations to recover. And while a strong native seed bank is critical for the health of these species, a persistent invasive seed bank can make eradication goals extremely difficult. But we really don't know a lot about seed banks and which species maintain them. So we can think of seed banks as a method of temporal dispersal. It's a way for species to disperse their seeds through time, spreading out germination across multiple years due to either high seed survival or high dormancy, or a combination of the two. Whereas other species may focus more on spatial dispersal. So utilizing vectors like wind, water, and animals, including humans, they're able to scope out new territory. But how can we predict whether a species spreads spatially or temporally? Well, the first place I like to start when trying to figure out how species cope with and respond to their environment is by looking at functional traits. So functional traits can tell us what plants do when they're faced with limited resources. There are these inherent trade-offs that species make. So, for instance, plants trade off between growth and water conservation. And one functional trait that's really well studied and correlated with this trade-off is specific leaf area. So species with large, thin leaves are more efficient at capturing light, helping them grow quickly, while species with thick, small leaves are better at conserving water, but grow more slowly. But I'm really interested in whether species trade off between colonization via spatial dispersal and temporal dispersal via seed banks. And are there traits that underlie this trade-off to help us predict these functions? And to understand dispersal, we really have to look at seeds. Seeds are responsible for maintaining current populations of plants and establishing new ones. They're how plants move around to find new patches to colonize. And it's through their traits that we can begin to understand how, where, and under what circumstances they're able to do this. So for my talk, I have three main questions that I'm going to be addressing. First, how do seeds vary in their functional traits, and do these traits differ between natives and invasives? And I'm focusing on annual species here. Second, whether natives and invasives differ in their abilities to disperse spatially and temporally, focusing on annual grasslands. And finally, whether seed traits are correlated with a species' ability to disperse spatially and temporally. For the first question, I'm just going to be looking at traits. For the second question, I'm going to be using some modeling. And for the third part, I'm going to be combining the two. So first, to assess how functional traits of annuals vary, I really wanted a large range of seeds from annual species. So with the help from California Botanic Garden and the USDA and many other collaborators, I've obtained seeds of 300 species, including 250 natives and 50 non-natives. And over the past year and a half, we've been measuring a suite of functional traits on these seeds that are related to temporal and spatial dispersal. And here I really have to recognize this amazing group of undergraduates that I've been working with on this project. They've dedicated countless hours to measuring these traits in the lab, and they've just been overall wonderful to work with. All right, so to then estimate each species' ability to disperse temporally and spatially and link those with the seed traits, I'm utilizing these 5 long-term data sets from across my range of seed collection. But today, I'm just going to be focusing on this one at McLaughlin Natural Reserve. This is the site that we see the highest diversity of natives and non-natives. So McLaughlin is a UC Natural Reserve in the coastal foothills overlapping Lake Sonoma and Napa counties. It's a typical annual grassland, but also has really large patches of serpentine, leading to high native diversity in addition to the typically abundant annual invaders. This data set of 80 sites has been monitored by Susan Harrison for the past 22 years and includes enough data to estimate spatial and temporal dispersal in 60 species. And to estimate each species' ability to disperse temporally through the seed bank or disperse through spatial colonization, I'm utilizing these models that are called hidden Markov models. So hidden Markov models take long-term presence-absence data on a species as seen above ground to predict its presence below ground, and then we can estimate transition rates between those two states to extract each species' ability to disperse through space and to disperse through time. These models were developed in the animal behavior and movement fields to account for the fact that during a census, you might not see every individual, but that doesn't necessarily mean that there's been a mortality event. This is just like a seed bank, right? So seeds of seed banking species may survive for years before germinating, so even if we don't observe the plant above ground, it may still be present below ground. All right, so moving on to my results. So for the first question, again, just looking at traits. And we're still in the process of measuring traits, but this is a principal component analysis of all the traits we've measured so far. So we see seed shape, size, mass, seed release height, settling time, wing loading, nitrogen content, and carbon content. So we can look at how these traits relate to each other and drive variation among species. And you can see that differences in mass are really driving the majority of variation in these species. So it's really highly correlated with PC1. So species with high PC1 scores have low mass, and species with low PC1 scores have high mass. But PC2 explains this trade-off between species with larger, more elongated seeds with low nitrogen content and species with smaller, rounder seeds with high nitrogen content. And what we find when we look at how all 300 annual species from across my collection range are distributed across this trait space is that we actually don't see much of a difference between natives here in gray and non-natives here in green. But when you account for the Calypsy rating of invasiveness for these non-natives, we start seeing some patterns. So in this ordination, natives are still gray, but now the highest impact invaders are in green, moderate impact invaders are in purple, and those with limited impacts are in orange. And then the yellow ones are species that haven't been assessed yet. So there are some stragglers, but for the most part, those species with the highest impacts, those in green and purple, have larger, more elongated seeds. Okay? All right, now we're going to look at the McLaughlin species in particular and their spatial and temporal dispersal abilities. So this is a graph of hidden Markov model estimated spatial dispersal in native forbs, non-native forbs, and non-native grasses. And you can see that the native forbs in purple overall have lower spatial dispersal abilities than the grasses in yellow, but non-native forbs in green don't really differ from either group. Similarly, when looking at temporal dispersal, we see native forbs, again in purple, have higher temporal dispersal abilities, so they're more likely to maintain seed banks than non-native grasses. But non-native forbs, again, don't really seem to differ from either group. Further, we do see a trade-off between these two abilities. So each dot here is a species within the McLaughlin data set. And species seem to be able to prioritize on either spatial colonization or temporal dispersal through the seed bank. Finally, if we combine traits and these models, are there seed traits that can help us understand which species are likely to specialize on spatial spread and which species are likely to maintain seed banks? First off, I'm not even going to show the graph, but there was no relationship between mass, which was PC1, and a species' ability to disperse spatially or temporally. Instead, we see that the second axis, PC2, which was correlated with species shape, size, and nitrogen content, actually does a pretty good job of explaining this trade-off. So species like Avena fatua and yellow star thistle are better spatial dispersers and have larger, more elongated seeds with low nitrogen content, whereas species with smaller, rounder seeds like Lepidium nitidum and Gilia tricolor are better temporal dispersers. This suggests different management strategies for weeds and natives at different ends of the spectrum. So for weeds with strong spatial dispersal, it's important to coordinate management efforts across boundaries to manage propagular pressure, whereas species with higher temporal dispersal will want to spread resources and management efforts out over time to exhaust the seed bank. Similarly, when restoring natives, it might be best to focus on those that can spread spatially at first where populations are establishing, and then working to increase those with the seed bank. So some takeaways from this work: invasive grasses are better spatial dispersers, while native forbs are better temporal dispersers. Non-native forbs seem to be much more variable, but their traits can help us understand which species specialize on temporal dispersal and which ones specialize on spatial dispersal, because we do see that trade-off between these functions, and it can be predicted by their traits. So species with those elongated large seeds are going to be better at colonizing, while those with smaller, rounder seeds are better at maintaining seed banks. And that's it. I just want to recognize, I work with some wonderful people, everyone in the Grammar Lab and the Hallett Lab, and in particular, 3 undergrads who've really made this work possible: Michael Borton, Lauren Ward, and Kate Cox. Thank you.
Hello, hello, hello.
You have a list of some of those natives that have the better spatial dispersal?
Oh, yeah. Get in touch.
Have you mapped onto this with the native species, the indigenous uses for the whole?
I turned it down, yeah.
Okay.
Yeah, one lady is right in front of the speaker, so if I turn up, and that's why I kind of feedback a little bit. Okay.
That's a great question. So is mapping with indigenous uses for these species? I haven't looked into that, no. Do you know any good resources to do that?
Tending the wild, there's several books that are really good.
Yeah, I'll check that out. Thank
Can you repeat the question, or whatever he said?
Oh. The question was about whether or not I've looked at indigenous uses for the seeds that I assessed in a study, and I have not, but that's a good avenue that I'll explore. All right.
Just a quick question. For the hidden Markov models where you're calculating the probability of spatial and temporal dispersal, what were the units for those two, if there were any?
It's a probability. So the units are basically like the likelihood that a species partakes in spatial dispersal versus temporal dispersal. Yeah. Great. Thank you.
Wow, that was cool. All right, well, let's move on to our third speaker. Debra Ayres did her PhD on Wyethia reticulata, a rare plant of Pine Hill in El Dorado County, and then she worked for a long time for UC Davis, and now, as a retiree, she studies the rare species of Pine Hill and the Gabbro-Chaparral community dynamics, and is an active member of the El Dorado County chapter. Welcome, Debra, and correct anything if I missaid it.
Okay. All right. Well, I'm very happy to be here today talking about a process that began about 15 miles from where you're all sitting. In 1975, the U.S. Army Corps of Engineers decided to solve 2 problems they had: a subsided salt pond and an excess of dredge spoil from the Port of Oakland. So they dumped the dredge spoil into the subsided salt pond instead of out to sea, and decided to create a marsh on that elevated salt pond. Things went awry when a biologist for the Army Corps was able to find and purchase from Chesapeake Bay viable seed from Spartina alterniflora, native to the East Coast. He had an East Coast eyes and thought that this plant was going to be far superior than our native species in restoring that salt pond. And the Army Corps found that it was a tremendous recruitment site for Spartina. Spartina alterniflora is native to the U.S. East Coast. Foliosa is the native California cordgrass. Come here, stop that. Okay. Both of them are keystone species in their respective marshes because they can withstand inundation and salinity at the lowest tide elevations, forming the edge of the marshes. Spartina alterniflora is a potent ecosystem engineer. I can do this. Here it is in the East Coast. Sediments from tidal waters settle out, and the plant grows into those sediments and eventually elevates the marsh, you can see, over 1 meter in height. This is Willapa Bay, Washington. This, you can see an island out here that is a clone of Spartina, a single individual. These individuals coalesce and form vast meadows in both the native and the invasive ranges is, again, Willapa Bay, Washington. Spartina reproduces both by seed and by vegetative growth. It is wind pollinated, and in the native ranges, it is mostly outcrossing due to early acting inbreeding depression, which kills the embryos. So for a long time, it was thought to be self-incompatible in the genetic sense, but it is not. So it's obligately outcrossing due to this inbreeding depression. There is no seed bank, and the seed spreads on the tide. Individual seeds settle out in the open mud and grow equally in all directions, forming these circular clones. Hybrids were first detected in San Francisco Bay by Kurt Daler and Don Strong using 4 rapid markers, a type of genetic marker. And then I came on and developed some more markers and did the first Bay-wide map. Our mapping methods were initially to walk through marshes, collecting leaf samples every 10 paces or so, and then return to the lab. Later on, working with the ISP, the Invasive Spartina Project, they launched themselves in kayaks. So the way it works is that we developed a suite of molecular markers that were unique and ubiquitous to each species. There's alterniflora's band. Here is foliosa's band. So that individuals that contain the bands of both the parental species had to have arisen through hybridization. What we found in 1999 was a preponderance of hybrids in marshes where we found the hybrids, and these are the introduction sites, Alameda Island, Alameda Creek and Hayward. It was almost absent in the South Bay, absent in the North Bay. 5 years later, and with a lot of help from the Invasive Spartina Project, teams of technicians went throughout marshes everywhere and returned the samples to us for genetic analysis. You can see it now. Covered the South Bay, East and West Bay, and had proceeded to go into the North Bay. So to understand this, I want to present what a normal plant does when it invades. I don't know if that makes it normal. So this is Willapa Bay, Washington, Spartina alterniflora spread. And in order to portray this without having the graph go through the ceiling, we use a logarithmic scale and a linear time scale. So this is a log linear graph, and exponential growth is a straight line in a log linear graph. So the straight line is the intrinsic rate of increase R of that species and population. Why is there that box there? I have no idea. There shouldn't be a box there. We did a similar analysis in San Francisco Bay, again, log linear, and between every 2 dates, there was an increase in the growth rate, not a straight line. And we called this super exponential growth, or greater than exponential growth. So what's up with that? So these plants hybridized. This is a structure diagram, and I'll go into detail about this later. They are homoploid hybrids. They have the same chromosome count as the parental species, that is about 60 chromosomes. There is no speciation. And the hybrids apparently bred with each other. They back-crossed with the parental species. They had a genetic free-for-all out there in the marshes and produced this genetically diverse, what we call the hybrid swarm. It was bidirectional. Chloroplast DNA is inherited by the seed, from the seed parent, and so both species were parents to hybrids. So to compare the two bays I just showed you, we had a similar introduction into this open niche habitat. Open mudflat is the normal system. That we had hybridization in San Francisco Bay, no native Spartina in Willapa, so no hybridization. And what we found is that evolution was fueled by genetic variation and resulted in the accelerating spread that we saw. On the other hand, evolution was constrained in Washington by a lack of variation and resulted in the constant spread rate we saw there. So how does that work? The complementarity of genes from each of the parental species and strong natural selection results in hybrids that were greater than either of their parental species in these various traits, pollen production and viability, leading to pollen swamping. And that's when a single hybrid individual will effectively and efficiently cross-pollinate all the plants surrounding it with its vigorous pollen. There are higher flower intensity in the hybrids, higher seed set, including cell fertility, and I'll show you the evidence for that, and clonal growth. And this led to evolution in contemporary time. And by contemporary, I mean in about 25 years. And if you think that's rapid, just consider what COVID-19 has done in 2 years. What does that look like on the ground? So the upper picture is a native marsh at China Camp. You'll see Spartina foliosa, our native cord grass, is about the height of a great blue heron, and you can see that it's sparse. You can see the waters coming through. On the other hand, Katie Zarimba, who's 5'6", is demonstrating a hybrid invasion marsh just north of the San Francisco airport. To now go into a little bit more detail on the genetics of hybrids. This is a structure analysis, and you've probably seen it before. But what this analysis does is it takes the dimensionality of 17 microsatellite markers and all of that variation and boils it down into 4 populations that are genetically affiliated with each other. And each of these populations is represented by a different color. Oops. So Spartina foliosa is in blue. Spartina alterniflora from Chesapeake Bay was red. And surprise, surprise, we found a completely different Spartina alterniflora off Alameda Island, and we had some white literature that suggested the Army Corps had planted alterniflora there. Clearly from a distinct and different population. I'm voting for Louisiana, but anyway. Each of these bars is a separate individual. So we have an individual that's pure yellow, is alterniflora, and this guy here is a mixture of the 2 alternifloras. So this represents that genetic diversity I was talking about. And I want you to look here. These are seedling populations over 2 years, seedling populations. And notice how that all of these individuals look like each other genetically. There isn't this huge mixing that seed floating on the tide would seem to suggest, but rather that seedlings were locally produced and dispersed. I want you also to notice that somehow we pick up this green color. Where did that come from? Well, 30% of the individuals here came from this single plant, solid green, that was isolated out on the mudflat, and through self-pollination, it produced all of those offspring. So it regained or gained self-fertility by purging its inbreeding depression. How this usually works when alterniflora invades, and in fact it's been introduced a number of places, because of that self-infertility, the population has to wait until there's coalescence among these various individuals. Again, these circular areas are clones. This is a phenomenon known as the Ali effect, if you've heard of that. And then they produce seeds, which colonize the mudflat, and then the plants grow, and finally they turn into these seed-producing meadows. What happens in San Francisco Bay is that the hybrids are self-compatible. So there is no waiting around for a pollen partner, because you are the pollen partner, basically. It cuts a lot of time out of the process. So what is driving this invasion? Is it that pollen swamping I talked about? Is there higher seedling survival of hybrids? A higher growth rate of the clones? So we were lucky to have Richard Hall develop a mathematical model to evaluate those various hypotheses. And what he found, so this straight line, the straight black line, that is merely exponential growth. When you add in increased hybrid seedling survival, you start to get that greater than exponential growth. And increased clonal growth rate also contributes to that spread. Pollen swamping did not add to spread rates. The end result, if there were no control of the hybrids, is the extinction of both parental species. So this graph, this is time. At time 0, there was very few Spartina alterniflora growing in that salt pond from the Army Corps. The -1 is the foliosa, very abundant. But this rare invader, through hybridization, basically is able to overtake through genetic assimilation a native cordgrass. So we know that seedling recruitment drives this invasion, and recruitment is a function of various plant factors, which I've talked about, and also environmental factors. Safe sites for seedlings. The seed must arrive at a place where the seedling can survive, and a favorable climate, it turns out. So what do these safe sites look like, and what is the danger of a seedling on an open mud flat? It's tidal action. Tides come in, tides go out, and they rip out those seedlings. Anytime you can protect those seedlings is where they're going to survive. And this is probably the mother of these plants. It created a nice little island here, and in the lee of this island, it provided a safe site for its offspring. As the Army Corps found out, and as we found out in the 90s, restored salt ponds were tremendous sites for Spartina recruitment. This greeny-gray color is open mud, and this red are the false image of the plants. Restored salt marshes are graded. The tide is muted, so there's none of that ripping out of the mud. And back here in the 90s, the tidal channels were lined with hybrid cordgrass, which delivered seeds into the channels directly to the salt marsh. We also found something intriguing. We had a strong El Nino year here, and here's the aerial photograph. The consequence of that El Nino year was this tremendous spread of the Spartina in this salt pond. So we went out and we measured during high rainfall years and found that high rainfall promotes clonal growth in all the Spartina, but especially so in hybrids. Flowering and seed set, again, in all Spartina, but especially in hybrids. So they love that fresh water. When those seedlings that Christina and I collected and she genotyped, we walked mudflats looking for seedlings. And sometimes it's put out that there is no recruitment by seedlings, to which we say, oh, yes, there is. But it has to be during a certain year, and you have to walk miles of mudflat, and then you find them. Here, these are the 3 marshes that you saw the genetics of. Then following those El Ninos, and then the La Nina year, there was almost a complete cessation of seedling occurrence. So we conclude that seedling recruitment is episodic due to climate and dependent on sites, safe sites, which also tells us how to control hybrid Spartina, the elimination of all hybrids to stop pollen swamping and hybrid seed production. And you want to be especially vigilant during El Nino years when you can expect to see massive growth. And the reduction of safe sites, pilings, restoration sites. And right now you've got a project, the South Bay Salt Pond Restoration Site, which I kind of cringe. No genetic analysis has been provided on the cordgrass that is recruiting into those restored salt ponds in Alviso. So it's like, I don't know if you folks have done it, but scary. I mean, evolution doesn't stop just because we decide it has to. And of course, this affects more than just plants overgrowing mudflats. It influences the entire ecology of that ecosystem. And this is a nice volume available online that captures all the many and various ways that Spartina impacted the mudflats. Available from the Invasive Spartina project. And finally, I would like to thank Team Spartina at UC Davis, Professor Don Strong, who mentored all of us, and the various undergraduates in the lab, postdocs who came out, graduate students, and including June Bando, who's a grad student in our lab. And in this case, she was investigating barnacle colonization of a shopping cart. So I'm ready for questions if you have any. Oh, I have to say that the Invasive Spartina Project will be speaking next to tell you what they are doing now.
About 5 minutes.
Oh, my goodness.
Could you explain a little bit how the fauna is adapting to the new hybrids and kind of some of the ecological changes that you're seeing?
There have been extensive work looking at just how is the fauna adapting to this new hybrid swarm? Actually, it's been beaten back from over 880 net acres to something like 22. Evidence from Washington say that those shorebirds who wade out in the mud, they don't go into the Spartina. They know better. The trophic cascade goes all the way down to the invertebrates that live in the mud. So huge changes. And the people who board surf on the open mud, they were our allies too. They didn't like it. Any other questions? Okay.
Hi. Sorry. So why is it that you think that specifically Spartina reacts well to something like El Nino with a lot of freshwater rain, and specifically why are the hybrids reacting even more? Like, do you have any understanding of that yet, or no?
Well, sort of is more of a coincidence what those high rainfall, and it's twice the amount of rainfall as a normal year, brings down more sediments, and so there's more material for the plants to grow into, and fresh water. During the El Nino in '97, it was fresh water out to the Golden Gate. The plant, it doesn't require salt to grow, but it's one of the few plants that can grow in the salt. So it's got an advantage where there's brackish water or saline water, but it likes the fresh water. And so it performs better. And why does the hybrid perform better? Because it does everything better. And I want to say there isn't just one hybrid, there's a hybrid swarm. So there are hybrids out there that are puny and unfit, and they died. But there were hybrids that were robust and tall and productive, and they're the ones that were selected by natural selection to be the survivors. And of course, those genes then went on and on and on through future courses of natural selection. That's how evolution works. Okay.
You mentioned all the genetic differences. Is there any physical differences between the plants that are noticeable? Or?
Oh yeah. We grew them in a common greenhouse environment early on, and we measured the various traits of all these types. The ISP knows what they look like in the field. The hybrids, 70% hybrids, according to our markers, were the most fit. Not alterniflora. They beat alterniflora, but they also have red culm color down at the base, which you don't find in the native at all. And so these observations have been, I think, somewhat confirmed over the years through genetic assay of these suspicious plants. Yeah, so we can recognize them. Although there are cryptic hybrids that are sampled, they look like foliosa, but they're hybrids. So we got cryptic hybrids also in the mix, these littler plants. Okay, so our next speaker is from the ISP, and I'm so excited.
Thank you, Debra. This was a great opportunity for us to hear from one of the folks that found out about this original hybrid situation, to hear about the Spartina, the hybrid Spartina story, and about this pretty much now a textbook case of the evolution of increased invasiveness through hybridization. So thanks a lot for that. Important thing to consider. Our next speakers here, we have a wonderful ability here now to get the beginning and the current situation. Our next speakers are Jeanne Hammond and Brian Ort from the Invasive Spartina project. And Jeanne is the Restoration Program Manager for ISP, and Brian Ort is the Genetics Program Manager for the same project. They're going to be doing a team presentation, and a lot of this work is funded by the State Coastal Conservancy, so it's been a wonderful example of a very complex project to deal with a very complex problem. Thanks. Oops, did I just mess you up? Okay. Is this still yours? Yeah. Ah, okay. Got it. Here you go. I'll just go the normal run.
All right. Thanks, Debra, that's a great talk and a great introduction to what Jeanne and I are working on. We both work for Olofson Environmental as part of the Invasive Spartina project, and we're going to talk about our efforts in restoring tidal marsh and mudflat habitats in response to this hybrid Spartina invasion. So tidal marshes used to fringe pretty much the entire San Francisco Bay, and they are important habitat for a large number of animals and plants, including these rare, threatened, and endangered species I have listed up here. And they also provide us with a lot of ecosystem services like flooding protection, but they're threatened. We've lost about 85% of the tidal wetlands that existed in San Francisco Bay in the last 200 years due to human development, and currently even more threatened by effects of climate change, like flooding, just being flooded out. Debra introduced the topic of the introduction of East Coast smooth cordgrass in the 1970s, which then hybridized with our only native Spartina, Pacific cordgrass. And what I want to point out or emphasize is Pacific cordgrass, or Spartina foliosa, typically would only occupy this narrow elevational niche within a native marsh, from the upper edge of the mudflat or the upper edge of a channel up to the lower part of the middle marsh, which we see as the pickleweed plain. And as Debra said, hybrid Spartina can occupy that entire elevational range and come to dominate it and crowd out and outcompete all of the native plants, or any other plant, native or not, that occupies that marsh. And here's a little bit of it in action. As Debra said, it is an ecosystem engineer. And what it does when it gets out into the mudflat is it changes changes the mudflat environment for the invertebrate community, which now, instead of living in mudflat, is trying to persist in the rhizomatous mass. Yes?
That plant right there was the sulfur that produced all of the seeds, just so you can see how extremely isolated it was. Sorry.
Yeah, no, thanks. That's great. That's fantastic. Now I don't have to make that point. No, it's absolutely right. So this is great because this is in 1986, the mouth of San Lorenzo Creek. This is the same photo, 2004, and you can see that the few clones that existed in 1986 have now probably, I'd say, covered, a tenfold increase in area of coverage. And that's a monoculture of hybrid Spartina. And what I was going to say about the invertebrate community, it totally changes the invertebrate community. What you now have are invertebrates that live in rhizomatous masses and not mudflat inverts that the birds like to feed on. Of course, that affects our ecosystem services and eventually will cause the failure of any marsh restoration that you're trying to do anywhere in the bay unless you address this issue of colonization and spread by hybrid Spartina. So in response to this threat, the Invasive Spartina project was organized in about the year 2000, fully implemented in the year 2005, and it is co-directed by the state Coastal Conservancy and the federal U.S. Fish and Wildlife Service, with management and implementation by Cal-IPSI and us at OEI. And it's composed of 4 different programs. Inventory, where we biologists go out and traverse all the marshes, and we map invasive Spartina, hybrid Spartina, wherever we find it. And yes, mostly we can do it by morphology. Where we can't do it by morphology, we do have a genetics program. I'm the geneticist. I don't get to talk about genetics today, unfortunately. But we go out, we map it everywhere we find it. In the treatment program, we lead treatment crews back to it, and we spray it with an herbicide called imazapyr, an approved aquatic herbicide. And then when it's appropriate, we can then implement the restoration program with habitat enhancements, especially focused on this guy, the California Ridgway's rail, which is an endangered bird that actually likes the shelter provided by invasive Spartina, although it feeds on mudflat. And then we monitor rail populations, and that goes back and informs all of the rest of our programs every year. This is what inventory monitoring looks like. We have 2 biologists out here carrying handheld, GPS-enabled tablet computers. They record hybrid Spartina wherever they find it. If they would like to take a genetic sample, they can do that, and we can ID it based on lab results. And they're out there traversing up to 70,000 acres a year, June to November. This is what one day looked like back in August 26th. 12 biologists went out and traversed, this is Ogilvie Island here in the South Bay, mapped hybrid Spartina. It shows up red when we map it. When we lead the treatment crew there, we update the symbology to blue. So this is from September 15th, treatment day. And what I want to point out, there's a lot of hybrid Spartina mapped here. And it looks bad, and it is an infested island. But almost all of the space in between what's mapped is Spartina foliosa, Pacific cordgrass. So I want to point out that this is a Pacific cordgrass island infested with hybrid. That's important to not be misled. We don't map Pacific cordgrass, so it doesn't show up on our maps. We've been very successful so far. In 2005, when we began, we had 805 net acres of hybrid Spartina. We are now down to 22.5 net acres. We're proud of that. We're also a little bit single-minded. We want to get the last 3%. And it's important that we do, because, as we said, any infestation will ruin a restoration project if it's not contained and controlled. We do have several sites that are suitable for restoration. Out of an initial 227 sub-areas that were infested, we now have 54 where we do not detect any hybrid Spartina, 53 more where we're down to less than 1 square meter, 48 more that are less than 10 square meters. And this is ripe for restoration, which now I will hand over to Jeanne.
Thanks, Brian. So why do we have a restoration program? maybe that's already obvious to you guys. As both Brian and Debra talked about, invasive hybrid Spartina has caused a lot of negative effects on our tidal marshes, our mudflats, and on ecosystem function in general in the San Francisco Bay. And one of the main reasons for this, they've also talked about, is that hybrid Spartina can grow in a much wider range of tidal elevations than our native cordgrass. So that means it grows down into mudflats. And as you can see in the slide here, it can grow, it colonizes, and then it spreads really rapidly, and you end up with no mudflat. Well, that's a really big problem because a lot of creatures use our mudflats as really important habitat. Migratory shorebirds, waterfowl, a lot of wildlife. In addition, hybrid Spartina can grow up much higher elevations onto the marsh plain, and there it outcompetes, as Brian said, it outcompetes the native vegetation. So we end up with a loss of biodiversity, and we end up with not having that mosaic of habitat types we expect to see in tidal wetlands. What it did do, however, like Brian said, is that it provided novel habitat where there was no habitat before. So this is a really good example of this is our early restoration marshes, where we breached and restored to tidal action, and you would expect that passively native marsh species would recruit into these sites. But if hybrid Spartina was nearby, that didn't happen. Hybrid Spartina came in and it became a monoculture, a meadow of hybrid Spartina. Well, ridgeways rails learned to use that habitat. It's good habitat. It's habitat. There was mudflat or a restored salt pond before. There was nothing there. And therein lies the challenge for our program. We're trying to control hybrid Spartina, but we also need to protect and preserve ridgeways rail habitat. So the goal of our program has been to restore tidal marsh habitat for the benefit of ridgeways rail and other tidal marsh dependent wildlife. So our program's been going on for 11 years, and we're currently planning our restoration efforts for this coming winter. We have a plan that guides all of our restoration efforts, and that's informed by a technical advisory committee. We work on a bay-wide scale, obviously, and we focus on key components of rail habitat. Birds need cover from predators for foraging, nesting, and for refuge from high tides. And we focus on rapid enhancement because we want to get in there and provide habitat for the rails as quick as possible, and also because we know a lot of these young restoration marshes need to be on a faster trajectory towards mature marsh because of the threat of sea level rise and climate change. So how do we and where do we do our enhancements? ISP and partners have worked at over 40 sites around the bay. We've planted over 500,000 Pacific cordgrass, marsh gum plant, and various transition zone species. And we've also constructed 82 high tide refuge islands at 16 sites, and I'll be talking about that in a little bit. The graphic on the left shows we worked mostly in the Central and South Bay. And as Debra talked about, this is the original introduction site, so you can imagine that the impacts of invasive Spartina have been stronger in these areas. So what do we plant? I've included the marsh zonation diagram that Brian included in his part of the talk here to show you where in the marsh zones we're putting these species. So Pacific cordgrass, like Debra talked about, is the only species in San Francisco Bay that really can grow at low elevations. And it's a really great cover for foraging for ridgeways rail and other wildlife. In the marsh plain, just up here, we plant marsh gum plant, and that's to provide additional taller cover for rails along channels. They use the channels a lot in the marshes. And then the transition zone is the space between the marsh plain and the adjacent upland habitat, and that area is really important during extreme high tides. So I talked about the fact that we need to do our enhancement rapidly, both because we're trying to provide habitat for rails quickly and also because of sea level rise. So our planting designs are high density, they're grid patterns, and that's because it's easy and quick for contractors to install, and we can use that same planting design and repeat it across that same site or across sites, and it's just very repeatable and easy to use. Example in the middle is of one of our designs is for marsh gum plant, the marsh plain species. And the idea here is that it's 2 plots on either side of a channel, and each one of those plots has 20 plants planted very densely. And it's creating a node of habitat. So you can envision a ridgeways rail pair using that channel for foraging. A predator comes and they need somewhere to hide, so they hop out of the channel up into the gum plant. And then you repeat these nodes of habitat across sites that don't have this additional cover. And this is a slide showing before and afters of some of our sites. So up in the upper left-hand corner is an early restoration site at Eden Landing Ecological Reserve. You can see it's not vegetated yet. It's still at early stages. So we wanted to come in there and rapidly plant it. The thing you should know about this complex is there's no local source of Pacific cordgrass here. So we came in and planted rapidly, and you can see habitat cover on the right-hand side. And the two sites on the right are both sites at Hayward Regional Shoreline. You can see they have passively revegetated really well, perennial pickleweed. But what perennial pickleweed doesn't provide is that taller heterogeneous habitat cover that you need for rails. So we came in and planted, and you can see on the right-hand side the result of that. And then on the lower left-hand side in the transition zone, this is a space we've just started working in this past winter. And the idea here, remember, is we're trying to provide some place for wildlife to go when those king tides fill the marsh plain. So we're planting perennial species that are going to provide year-round habitat cover. And the other type of enhancement that we've been working on in collaboration with H.T. Harvey and Associates and contractors is high tide refuge islands. So USGS and other researchers found that ridgeways rails, their highest mortality happens during the winter when we have the king tides or the extreme high tides, especially when they combine with storm events. And so a lot of our marshes, when you're out there, they don't have any high tide refuge in the middle of the sites. So the idea of this is it's a very small habitat feature, but you can build a bunch of them and put them at different places, especially along channel confluences. So you can see this example one here, there's channels. So the rails use these for foraging, and then they can hop up on the island when everything goes under the water. These have been successful, and this next slide shows two different locations where these islands have been constructed. And these slides are not showing a king tide, they're just showing a normal high tide. But you can see that even at a normal high tide, they're providing a lot of additional habitat cover above that water level. This is an example from Eden Landing Ecological Reserve, sort of the bigger picture. This is a salt pond that was restored to tidal action in 2005, and this photo is showing 2011, and you can see that it has not started vegetating yet. There is no local source of Pacific cordgrass here, so we did a bunch of planting. And you can see a couple of locations where we had photo points that I've put on here. Before and after, mudflat, and then now there's Pacific cordgrass providing habitat cover. And the graphic on the right, the green polys are where we mapped on the ground our plantings as they were expanding. We're no longer able to map out here. It's expanded so far beyond what this graphic can show you, but I put it on here anyway. And what I wanted to show you is that we've gone from the restored salt pond on the left to the little bird icons on there are ridgeways rails. So I don't know if you can see these guys right here. As Brian said, we do ridgeways rail surveys every year to inform the rest of our program. And so they've now come back in here and we detect them every year. So it's a really great success story, taking a salt pond, rapid planting, and we've got ridgeways rails there now. And this photo is just aerial imagery of the same site. This was taken in 2019, and the vegetation out there has expanded a lot since then. So this is another example of the introduction site. Once again, Alameda Flood Control Channel, and I just wanted to show you this again to show how incredibly invasive this plant is. You see the red circles and the blue circle on the left, those invasive Spartina clones are just starting to take over the mudflat, and by 2005, the mudflat's gone. And this is what it looks like now after successful control and extensive replanting of Pacific cordgrass. Once again, we have that narrow, continuous band of native Spartina that we want to see in a marsh in that appropriate tidal zone. It's not invading the marsh plain, it's not invading the mudflat. So the Invasive Spartina Project supports the Baywide Restoration community goal of restoring 100,000 acres of tidal wetlands. ISP also ensures that new restoration projects are not going to get invaded by invasive Spartina. they would become monocultures once again, like I talked about. That protects the mosaic of habitat types that we hope to see in our native tidal wetlands, and it also protects biodiversity. So our ultimate goal, of course, is healthy and diverse native tidal wetlands that support native flora and fauna. And that's it. So thanks. We have time for questions? Yes?
I think we're just about out of time here, but we will, I think Jeanne and Brian were happy to stay on for questions. So please, let's just continue on with a couple questions.
I thought Eden Landing was inundated with hybrid cordgrass and owned by California Fish and Wildlife. And go in there and then you eradicate the entire line, which is why you set up there left.
Exactly. Yep. Oh, so Debra is saying that she thought Eden Landing was completely invaded by hybrid Spartina, and that's true. Some of the sites there were completely invaded, and we've successfully controlled it. It's gone. Little bits of it here and there, but for the most part, it's gone. If it's a hybrid that's showing itself, we say that it looks kind of like corn. It's, it can be really tall. It's very robust. There isn't a lot of this left because we've gotten rid of all that hybrid Spartina, but that was the classic way to tell it apart. Our native foliosa grows about this tall, although in the South Bay it's much taller, but it's very even and it stays in its lane, and the hybrid is just tall. And like Debra was saying, it has red culms sometimes, purple leaf collars. There's a bunch of different traits that we can use to identify. Yeah, I mean, not always. So it's something, Spartina ID is definitely for someone who knows what they're doing.
It's a little bit like, the way I look at it is, if you're thinking about dogs, if somebody comes up and asks you, what does a mixed breed dog look like? It depends on what its parents are. I mean, there are many breeds of dogs, so it's not a great analogy. But in a lot of ways, what we're looking for is something that doesn't look like Pacific cordgrass.
Yeah, or something that doesn't fit.
I understand.
Right?
Yeah,
like
yeah.
A lot of the sites we work in are 99% native cordgrass. If there's something that looks different.
So in tandem with that, are you doing anything with Phragmites?
We would love to. I'm sure we all would, right?
Not in our mandate.
Not in our mandate, no.
How did the two species and the hybrid compare in terms of sediment accretion and relevant to climate change and sea level rise?
Well, as Debra said, the Spartina alterniflora on the East Coast is an ecosystem engineer, and the hybrid is an ecosystem engineer. So
it accretes more.
Mm-hmm. Yeah.
That's what I was seeing in the diagram.
Right.
I think that is at least in part due to the fact that it spreads so quickly, so it's able to expand its area of accretion. So it's fast. So
it's not all good either.
I know what you're getting at.
Yeah.
Who you got?
We got one more.
Else?
Yeah, who your call.
Go ahead.
So some wonderful work. What a great success you guys have made. I'm really shocked because I got involved early when this thing happened and didn't keep on with it, so it's nice to see something happen. But my question relates to how do you have an assurance that the foliosa you're planting now is not polluted with some minor part of genetics that might, show up later in time? How do you have an assurance that you're genetically pure? That's my first part of the question. And the second one is, well, never mind. Just answer that one. That's a big enough question. My mind's going off on too many things.
We did not get to talk. I mean, how do you fit everything into less than 20 minutes? We didn't talk about that at all. We have a very extensive protocol that we follow. We have propagation beds at the watershed nursery, actually, where, most of our collection happened 2015 and earlier. We use the beds to re-amplify each year, so we save a little bit of plant material, and then it grows again, and then we just keep. Before we ever collect something from a native marsh, we do a visual survey, we do an extensive grid sample of DNA samples, and Brian can talk to that. We also have like a criteria that it can't be anywhere near where we've ever seen invasive Spartina. So our current population sources are all North Bay because there's no hybrid up there.
And then spatially, you know they're far enough that you're not going to pollute from the Hammond.
Correct, yes.
And we continue to take samples and test the nursery beds.
Yeah, even though we've had no new inputs, we just do anyway.
Thank you.
Yeah.
Anybody else?
Thank you all. I'm sure that our speakers will be around for the break, all of them, and we hope to see you again tomorrow. Thanks. Bye-bye.